Okay, here we go. Thank you very much. So I will start from now. So the title of my topic is research is sex difference in the progression of glucose metabolism dysfunction in Alzheimer's disease. Since Professor Mook Jung really explained very well about the Alzheimer's disease and sex difference, so I briefly introduce about the Alzheimer's disease. So as you can see here, the major pathological hallmark of Alzheimer's disease is amyloid beta protein and tau tangle. So however, as you can see this disease stage graph, although there are small difference in amyloid beta burden, but it could become amplified in downstream processes such as tau accumulation or clinical dysfunction. So as you can see here, there can be the difference between the female and male in the progression of Alzheimer's disease. So as you can see these slides, there are, according to the stage of life, a woman can have some hormonal changes and also the changes of the brain glucose metabolism. So these two factors can be the potential link with the AD risk factor. So as you can see here, changes in sex hormone and glucose metabolism can be a potential link with Alzheimer's disease, but actually there are no direct evidence for metabolism and sex differences. So that's why I start to study this study. So the goal of this study is, the first aim is to investigate direct evidence of link between sex and glucose metabolism. And second thing is to find the blood-based biomarker for glucose hyper or hypo metabolism in Alzheimer's disease. And third one is to identify candidate genes linked with glucose metabolism in Alzheimer's disease. And the last one is to identify the discrimination power of these biomarkers for glucose metabolism in female. So this is the overall design of this study. The cohort 1 have 181 participants. It is the cohort from the Seoul National University Hospital. And we performed the Luminex and Simoa assay for the plasma amyloid beta and the plasma tau. And also we performed the PIB PET for the amyloid beta levels in the brain and the FDG PET for the glucose metabolism levels or neurodegeneration level. And we perform also the MRI for the cortical thickness and hippocampal volumic changes. So using this data, we perform the monotone regression spline curve analysis to mimic some progression of the disease, and also the partial correlation analysis, and also the RS curve analysis as well. So by using cohort 1, we try to find some the clue to whether some non-biomarkers can correlate with the brain, and it maybe we thought that it can be the different between the male and female. And for by using cohort 2, we try to find some female-specific glucose metabolic genes in Alzheimer's disease. By the publicly available database, we performed RNA sequencing analysis, and we get the data from the string database and the GO and RS curve analysis as well. So first, we need to check our cohort is really have different tendency between the male and female. So as you can see here, we could find the monotone regression spline curve is very different between the male and female. So as you can see, the right table, the comparison of spline slope analysis is very significant between the male and female. So we can confirm this data first, and we can do further analysis from this data. So secondly, for more detail, we need to perform some correlation analysis and the chi-square test and something the medical statistic tools. So as you can see here, in the female, the change of the FDG PET SUVR value, that this is the value of the brain glucose metabolism, is only significant in the female, significantly different in the female. And also, as you can see, the chi-square test, the female have more portion of the glucose metabolism dysfunction rather than the male. And we performed the partial correlation analysis with correction of age and the education levels. As you can see here, the female only have the significant correlation between the brain amyloid beta and the change of the glucose metabolism. But as you can see here, the number of patients is slightly different, so the reviewer actually requests to match the number of the patients. So we performed a randomized selection for females, and after we match, although we match the male and female number, we still can have the significant level of the p-value in the female only. So this means that there are sex differences between the glucose metabolism versus the amyloid beta burden. So we try to find the non-plasma biomarker can represent the glucose metabolism in the brain. So as you can see here, interestingly, the plasma amyloid beta 40-40 ratio is highly associated with the FDG PET data, which means the glucose metabolism levels. And also, we perform the correlation analysis, and we also get the female-specific data. So female has the significant correlation between the amyloid beta ratio, amyloid beta 40-40 ratio, and the brain glucose metabolism levels. So we can say that plasma amyloid beta 40-40 ratio can represent the glucose metabolism imaging data. So far, we had tried to find the non-biomarker correlation with the glucose metabolism. Then next, we need to find some specific genes, especially for the female. So we downloaded the GEO brain postmortem transcriptome data. So it has the 78 participants. And interestingly, just the differentially expressed gene is already very different between the male and female. So female has much higher levels of DEGs, as you can see here. So by using this, we are trying to find some specific genes for the glucose metabolism. So as you can see here, this is the reactome database. So from the reactome database, we sorted out the metabolic genes and glucose metabolic genes and try to overlap this transcriptome DG data and this metabolic gene data. So interestingly, the male only have one gene, but female has 26 genes. So it means that female differentially expressed gene from female has much more genes rather than male. So this is the list of the gene. So in this figure, we identify the female-specific glucose metabolism genes. Maybe it is the 25 genes from here. So next, we try to find the more specific genes for Alzheimer's disease. So I performed the string protein-protein interaction analysis. So I put in the 4 specific AD genes, such as APP in base 1 and 2, and MAPT, which can represent the amyloid beta and the tau. So interestingly, there are 4 genes are directly linked with this AD amyloid or tau related genes. So by using this, we can perform the the ROSCOP analysis with the integration of this gene by the largest regression analysis. So as you can see here, there are area under the curve over 0.88. So it is actually a very high level of the accuracy. We can get this kind of data. So the figure I is the summary of the data. So we perform the blood and brain, and for the blood, the longitudinal analysis, for the brain, and transcript analysis as well. So, okay. So in summary, in conclusion, our longitudinal and transcriptional analysis clearly reveal the sex differences in human metabolic changes. And also, by 2-year follow-up cohort, we can reveal some specific biomarkers such as plasma amyloid beta 40 to 40 ratio is highly associated with the brain glucose metabolism in women. And next, we identified the 25 novel female-specific glucose metabolic genes. And also, we find the four key attractor of FGG, female-specific glucose metabolic genes, like this. So, by using this kind of data, we can offer the crucial evidence on sex difference in the pathology of Alzheimer's disease. Okay, so that is the presentation over. But actually, I have the dual mode of my research. The first thing is the clinical application of the participants and the analysis of some medical statistics. And the next one is actually brain organoids. So I just want to show my research field just briefly. So I'm going to study also about the brain organoid for the mechanism study or the disease modeling. So if you have any question, you can also contact me as well. Okay, so this is acknowledgement. So today's talk is actually almost about the studies with the professor in Mukjang. So I really thank you about this project. And also we get the samples from human samples from the Seoul-National University Hospital by Professor Dong-Young Lee. And also this is the funding disclosed. And there's many collaborators. And this is our lab member. Thank you very much.